Okay, good afternoon, everybody. Today we're going to cover topic 16. So topic 16 is on translation. So as I've mentioned at least a couple of times, we have three big concepts that we need to know very, very well for the final exam. Replication, which we covered already. The second one is transcription, which we're going to talk about today. And the third one is translation, which we'll talk about probably on Wednesday. So let's get into this. Good news about transcription is that it's a lot more simple than replication. It's a lot more straightforward. So let's get to this. So first thing to talk about is transcription is where we're going to make RNA. And so what is RNA? RNA is ribonucleic acid. And how is RNA different from DNA? Well, first of all, it's usually single-stranded. There are some exceptions. There are some versions of RNA that are double-stranded. I think I mentioned last day the transfer RNA has some double-stranded regions, so there are some exceptions, but kind of generally we can say it's single-stranded. So what else? We can also say that the sugar is different. So this is where the R in RNA comes from. R is from ribose, right there, ribose. D is for deoxyribose. So if you take a look at RNA, RNA has an alcohol group right here at the second carbon, whereas DNA does not have the alcohol group, so we say it's deoxy, it's missing the oxygen, so that's where the D for DNA. Technically, it's 2' deoxyribose would be the full proper name for the DNA carbohydrate. On the bottom, you can see the other exception is that we have uracil. So the nitrogenous base uracil is found in RNA and not in DNA. In DNA, we have thymine. You can see the difference is thymine has this little methyl group. So slightly different, just enough that it helps the enzymes determine whether they're dealing with RNA or whether they're dealing with DNA. So here's an image showing all 4 RNA nucleotides. So you've got A, U, C, and G. You can see it's connected exactly the same way DNA is connected, except for there's a single strand. And the other exception, like I said, is the difference in the carbohydrate. So as I mentioned, there are 6 types of RNA we are going to be covering for the final exam, and you must know them. There's going to be a 6-mark question on the final exam. I haven't decided what the question is going to be yet. Last year, it was a fill-in-the-blank question where I had the name and the description, and you had to fill in what was missing. So sometimes you had to fill in the name, sometimes you had to fill in the description. Could be matching, could be fill in the blank. I haven't decided yet, but you should know these 6 types of RNA. So we've already talked about telomerase RNA, so that should be one word there. I guess it just got bumped over. And today we're going to be talking about messenger RNA and something called small nuclear RNA. And the other types, we're going to get to them, just not today. So make sure you know your types of RNA. So let's talk about RNA polymerase. So sometimes it's called RNAP as the abbreviation. And so this is kind of like DNA polymerase. What it's going to do is it's going to read a nucleotide template and it's going to make new nucleotides. So it's very similar to DNA polymerase in that it's going to make the new nucleotides in the 5' to 3' direction. But a couple of notes we can make about the RNA polymerase is, I'll make them on the screen here. So RNA polymerase, so come on, there we go. RNA polymerase. So it does not need the things that DNA polymerase needs. So it does not need helicase, single-stranded binding proteins, etc. It does proofread. It's just not as good of a proofreader as DNA polymerase, but it doesn't really matter. RNA are kind of temporary instructions. So if mistakes are made, it doesn't matter. The RNA is going to go away soon. The other thing is I will put a note and says it synthesizes in the 5' to 3' direction. So it synthesizes in 5' to 3' direction. So just like DNA polymerase, except for it's going to add RNA nucleotides. So add RNA nucleotides. So a lot of the principles are going to be the same though. But before we kind of get into how transcription works, I want to talk a little bit about exactly what it's doing around, because we have two strands of DNA, and so we got to kind of sort out what's going on there. So by the way, there are different versions of RNA polymerase depending on who you are. If you are a prokaryote, you have one type of RNA polymerase that does it all. And for some reason, eukaryotes have several types. So sometimes you might see in the notes something about RNA polymerase 2, right? So that's kind of the main one in eukaryotes. It transcribes messenger RNA, but there are different types of RNA polymerases. You can see that number 1 transcribes ribosomal RNAs. There's extra types found in plants. I have no idea why we do it that way, but it's working for us, so we're doing it, right? This is the way biology is. Sometimes we just do weird things. So like I said, I want to talk about the strands of these. Now I have this note here because usually I get to this slide and I draw it on the board. But what I've done is I've made a PowerPoint animation instead, and kind of want to talk about the two strands of the DNA. And this is why it's really important. Last day I was emphasizing why it's important that you know what the start codon is, because the start codon is going to tell us everything about these. So let's take a look at these two strands, right? You can see they're sitting there, two strands of DNA. They are anti-parallel to each other. One is 5' to 3', the other is in the opposite direction. So there's two strands. One we call the sense strand. Okay, so this is the strand that I guess we could say, I don't really have room to write on there. Maybe I'll write up here in the corner. Something about the sense strand is the sense strand has ATG. And so that's the start codon. So remember this, you may be thinking, but wait a second, isn't the start codon AUG? But remember, U is an RNA. In DNA, we're going to have a T. So this is kind of, you know, what we're looking for to identify the sense strand. So it's going to have the same sequence as the messenger RNA, except for the in the messenger RNA, all the T's are going to be converted to U's. So that's the sense strand, meaning that that's what we can read and it makes sense to us, right? The other strand, sometimes it's called the antisense strand. A lot of the literature, scientific literature calls it the antisense strand. I much prefer the term template strand. And why is it called the template strand? Because this is actually what the RNA polymerase is going to grab onto and it's going to read. So the RNA polymerase, wherever it sees a G, it's going to put a C in the RNA. Wherever it sees a C, it's going to put a G in the RNA. Wherever it sees a T, it's going to put an A. Wherever it sees an A, it's going to put a U. And so we'll see an example of this coming up. Kind of like the example I gave you last day where we did some transcription and translation. So two strands, right? It's important to know which is which. You're going to have a question on the final exam that's going to ask you to identify the strands. And the way you're going to do it is you're going to find that start codon for me. So I'll show you an example at the end of this lecture where we're going to do exactly that. So there's our messenger RNA. Like I said, it's kind of going to look like the sense strand of DNA. It's not going to be as long. We're not making RNA for our entire genome. We're just doing one gene at a time usually. So it's going to be, you know, maybe 500 or 1,000 nucleotides, and that's it. So something else to think about is that our RNA is going to encompass the gene. And it's going to encompass a little bit of DNA before and a little bit of DNA after the gene, right? So there's a couple of things that I want to talk about. One thing is this here called the promoter, okay? So this is worth writing a note for. So the promoter, so this is a DNA sequence. And it's before the gene. And this is where DNA, or where RNA polymerase binds. So you're probably thinking, why doesn't RNA polymerase just bind at, like, I don't know, the start codon? Well, you know, RNA is delicate. So we want to have a little bit of extra on each end, right? We can't just start at the start codon because you might miss you might miss some nucleotides. So we're just getting a little bit of extra just to make sure, right? So it's going to bind before the gene, and that area is called a promoter. So downstream is something kind of the opposite called the terminator. So this is kind of the opposite. It's also a DNA sequence. And this is where, and I guess I could write after the gene, where the RNA polymerase detaches. Okay, so I'm trying really hard with my handwriting. It's just very hard on the screen, so I apologize if it's a little bit messy. Those definitions are in the notes, the PDF notes that I gave you though. Okay, so these are all things to think about when we're going to make RNA. Okay, we're going to bind RNA polymerase before the gene. We're going to copy the sense strand by looking at the template strand, and then the RNA polymerase is going to come off at the terminator. So let's take a look at some of the details here. So we kind of break this into 3 steps: initiation, elongation, and termination. It just kind of means we're starting, we're making the polymer, and then we're stopping. So let's take a look at how this works. So there's my DNA. It has 2 strands, and RNA polymerase is going to recognize those specific sequences, and those specific sequences are called promoters. Okay, so there are sequences out there. Molecular biologists have catalogs of them. They know what they look like, and when we're looking for genes in databases, we're looking for these promoter sequences often. So there's the RNA polymerase. It's binding. And RNA polymerase does not need the helicase. It's going to unwind the DNA all by itself, and that takes a little bit of ATP. It's going to unwind that, and it's going to start making new RNA in the 5' to 3' direction. So here it goes. So the next step is called elongation, and that's just basically where it's going to continue along the way, along the gene, and make that make that RNA in the 5' to 3' direction. So you can see there's my new RNA transcript. So here's the 5', and it's going and sort of making the RNA in that direction like that. And it's going to keep on going. And like I said, what it's going to do is it's going to look at the template strand or antisense strand, right? So you can see here the textbook is now calling it textbook calling it template strand, so that's good. And in that case, like I said, it's matching complementary base pairs, right? Even though it looks like the sense strand, the enzyme is actually working along the template strand. So there it goes, it zips along. It's a pretty fast process, 10 to 20 base pairs at a time, limited only just by diffusion of nucleotides. So there's kind of a close-up, and you can see, so this here is the template strand labeled. And like I said, where there's an A, you're going to put a U. Where there's a G, you're going to put a C, another G and C. You see T, you're going to put an A, and so on. And so the RNA is made in the 5' to the 3' direction, right? So same thing as at this 3' end, that's the substrate, this thing here, and then these RNA nucleotides. So it's going along and making RNA, and then at the very end, the termination step, it comes along to the terminator. So it's kind of, it's going to be somewhere over here at the end of the gene. And the RNA polymerase detaches. And now you have a piece of RNA. And that's pretty much it. Like I said, relatively straightforward. We only have 1 enzyme to worry about. A few new words. I know promoter and terminator are kind of wishy-washy words. Geneticists tend to do that. They tend to make a lot of weird words that I don't like. But maybe you'll become a geneticist someday, and you'll like them better than me. Okay, so I have a little video here for you. It's kind of old. You can see 2001. It actually came with the textbook, and they haven't updated it. And so the graphics are a little bit weak, but it's short and it's sweet, and I think they do actually a pretty good job of explaining what's going on. So I'm going to play that for you right now. Here goes.
The first step in protein synthesis is transcription of RNA from DNA. The transcription of RNA from DNA begins with initiation. An enzyme called RNA polymerase attaches to a region of the DNA called the promoter, which lies next to the beginning of a gene. RNA polymerase pries the DNA strands apart, and RNA nucleotides line up along only one of the DNA strands, the template strand. RNA polymerase links the nucleotides together. The second stage of transcription is elongation. RNA polymerase moves along the DNA, adding nucleotides to the end of the growing RNA molecule. The RNA strand peels away, allowing the two separated DNA strands to come back together. Here's a close-up view of elongation. RNA nucleotides pair up with their complementary DNA bases on the template strand. Note that U in RNA pairs with A in DNA. RNA polymerase moves along the DNA strand, linking nucleotides to the end of the RNA strand. The last stage of transcription is termination. Once RNA polymerase has gone past a sequence called the terminator, the enzyme releases the completed RNA and detaches from the DNA.
So that's it. Like I said, kind of short and sweet and relatively good explanation. Just want to go back here and show you something that's kind of funny about this. This is prokaryotic transcription. So this is what is going on in E. coli and bacteria. And then, of course, for this this video, they have, I don't know why they have a nucleus. Has always bothered me, but not my fault. Somebody else's mistake.
The first step.
Oh, don't need to play that again. Let's talk about what's going on now in eukaryotes. There's just a couple extra layers of complexity here in eukaryotes. They do have a promoter, just like a bacteria, they have a promoter. So notice this promoter is often looks like this. It's often called a TATA box. So you're going to have TATA on one sequence, TATA, and the other sequence you're going to have AT. And so T and A only have 2 hydrogen bonds instead of 3, so it's kind of an easy place to break those hydrogen bonds and have the promoter start. It's really, really common in eukaryotes, so they have it in all these images here. So you can see there's the promoter, and we have a gene, you have a sense strand and a template strand. So all that is the same, just now you have this thing, this TATA box to think about. Now, there's one other thing that makes it a little bit more complex with eukaryotes, and this is because we are way more complex. If you think about, we've got many different types of cells. We've got heart cells, we've got skin cells, we've got liver cells, we've got neurons. So we've got different types of cells, and we have different developmental stages. We have a lot more DNA. We have those histone proteins to deal with. So what we have are something called transcription factors. And so these transcription factors help RNA polymerase bind. So some of them are kind of general. Some of them are tissue specific. Some of them are activated when we hit certain stages in life, like when you're a fetus or a child or hitting puberty, or from other environmental stimuli and things like that. So these transcription factors, it's kind of just important to know they're essential, right? They need to bind, and they're part of our genetic regulation. And then RNA polymerase can bind. So there's tons of these that have been characterized. Like I said, some are very specific, some are just a little bit more general. And so once they have bound, the RNA polymerase can come along. So notice it's RNA polymerase 2 in eukaryotes. And they form this thing here called the transcription initiation complex. So it's basically the RNA polymerase and all those transcription factors. And then everything else is basically exactly what we were talking about. It's going to make RNA. It's going to go along and read the template strand. It's going to add those complementary base pairs. Of course, it's going to be RNA this time and not DNA. And it's going to make that RNA transcript. There's an image from the textbook of a transcription factor, and kind of a cool looking protein, how it wedges itself there into the double helix there. Don't know a lot about transcription factors. Usually, sometimes they have RNA components as well, which is kind of interesting. So I kind of want to show you this image, and I kind of don't. I'm just trying to show you how complex things are. When it comes to eukaryotic DNA, there are promoters, yes, and then there's a whole bunch of other regulatory components. You can see this is talking about enhancers. There's also something called silencers and repressors and all these kind of things. I don't really want to get into those things, but I'm just showing you that things are very complex. And that's kind of the takeaway message. In terms of genetic regulation, we're going to talk about genetic regulation in topic, I think that's 16 or 17, and kind of just touch on some aspects of this anyway. So here we are back to our gene, and we have our promoter, and we have our terminator. I guess I wrote in an actual terminator sequence instead of the word terminator. And we've got our RNA, and notice I'm calling it pre-messenger RNA. So messenger RNA is going to code for proteins, but it actually requires a little bit of processing before we actually get to the point of where we're going to have messenger RNA in eukaryotes. In prokaryotes, the messenger RNA can be used right away, but in eukaryotes, like I said, there's some processing and other complications going on. So you can see it says here that there's a polyadenylation signal at the end. That's giving some stability to the messenger RNA. I'm not going to say anything more about that. What I'm going to do actually before I talk about the processing is I'm going to show you this quick video as well. This one is pretty cool. This is one of these graphical ones that shows you these animations, and I'll just play this for you now, and you can listen to what she says.
Transcription is the process by which DNA is copied into RNA in the first step of gene expression. It begins with a bundle of factors assembling at the start of a gene. That is, a linear sequence of DNA instructions. Here shown stretching away to the left. The assembled factors include an RNA polymerase, the blue molecule. Suddenly, RNA polymerase is let go, racing along the DNA to read the gene. As it unzips the double helix, it copies one of the two strands. The yellow chain sticking out of the top is the RNA, a copy of the genetic message. The nucleotide building blocks that are used to make the RNA enter through an
Oh, sorry, I don't know what happened there.
in the polymerase. In the active site of the enzyme, they are then matched to the DNA, nucleotide by nucleotide, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, thymine is replaced with the closely related base uracil, commonly abbreviated U. You are watching this process, called transcription, in real time.
Hey, so cool video. I thought you might like the sound effects, and kind of an interesting tidbit at the end, right? This is a real-time animation, apparently, which is just shows you how incredibly fast diffusion is at a molecular level. Okay, so before I talk about the processing and all that, I wanted to give you one of these sample problems. Okay, so you're going to have something like this on the final exam, so it's really important that you know how to do this kind of thing. This isn't the full question yet. This is, we're getting there. It's going to be a 5-mark question. This is probably, I don't know, maybe 3 to 4 marks out of the 5 marks, depending on how I decide to evaluate it. So it's going to look something like this. You're going to get a strand of DNA, and it says identify the sense strand and the antisense strand. So normally I do this on the board and get you all to do lots of writing. I kind of have it animated here today. It's just a lot easier. It's very tedious to write out A's, T's, and G's and all those kind of things and make sure that everything is correct. So it says identify the sense strand and the antisense strand or the template strand. So like I said, kind of the main thing to do is to look for the start codon. So if you don't remember what the start codon is, you can take a look at your genetic code, and it's going to be AUG. Of course, in DNA, it's not going to be AUG, it's going to be ATG. Oops, that's not a G. See if I can fix that. There, now it's a G. Okay, so let's take a look for ATG. I'm going to point out all the ATGs that I see. The one there, looking, don't see any more, and there's one here. There we go. So now the question is, which one is the right ATG? So this one here is not ATG. I know it looks like ATG, but remember in DNA language, we read things from 5' to 3'. So you're only looking for 5' to 3' ATG. This one here is actually GTA, because of course we have to read it in 5' to 3' direction. So the one on top there is the start codon. And so if you know where the start codon is, then you know which strand is the sense strand, because the one with the start codon is going to be the sense strand, and then therefore the other one is the antisense strand or template strand. Okay, so always find your start codon. This is what your DNA sequence is going to look like. I'm not going to have ATG at the beginning. It's going to be found somewhere in the sequence, and it's your job to actually find it. So then it says give the RNA sequence. So remember what I was telling you is that the RNA is going to look a lot like the sense strand, except for all the T's are going to be converted into U's. So let's do that. There's my messenger RNA. And I'm going to start right at the beginning, okay? I'm not starting at the gene because it's going to start in the promoter. And in fact, if you take a look, I actually even put in a data box here. So there's my promoter. So it's going to start somewhere around the promoter. So you're expected to basically transcribe the entire chunk of DNA, not just the gene. So you can see I've converted the T to a U and then copied down A and C. Going to keep going. TAT becomes UAU. The other thing you can do is just match complementary base pairs. I know some people have done this thing in high school, right? Where if you have an A, you put a U. If you have a T, you put an A, you put a U. So here I'm going to put another A, and then a C and a G. So that's another way to do it. And I'm going to continue all the way until I get right to the end of the gene. So hopefully that is pretty straightforward, right? At this point, you're looking at, that's probably 2 marks out of 5 there, right? Correct labels for sense and template, maybe 1.5 marks, not sure yet. I haven't written the question. 1 mark for the correct messenger RNA sequence, right? That you're doing the whole strand and not just where the gene is. And there you go. And so next it says identify where the promoter and terminator sequences are. So I've already showed you where the promoter. Remember, the promoter is going to be before the gene. So label some DNA before the start codon. And then where's the terminator? Well, we got to find a stop codon. So stop codons, you can see I have them highlighted there. So if we want to convert this to DNA, you're putting a TAA, TAG, or TGA. So the terminators are going to be TAA, TAG, or TGA. A little bit crammed in there, I know. And maybe you can see it. There's the TAA. So that means the terminator is somewhere after that. Okay? I'm not giving you specific terminator sequences, but I want you to label the DNA, the promoter somewhere before the start codon, for the terminator somewhere after the stop codon. Okay, and the next part, it says give the amino acid sequence. So what do we do here? We start at the start codon. So there it is. And usually what I do is just underline all the triplets I see, and I just keep on going all the way to the end until I don't have any more sequence left over. So at this point, it's just a matter of reading the genetic code and copying what I see. So AUG codes for methionine, UAU codes for tyrosine. So like I said, you can just use the codes that are on the genetic code. If you want to use the full name, that's fine. Depends on what I give you for the genetic code. So CCU is proline, and UAA is, of course, stop codon. So I'm done. This is the stop codon. I don't need to write stop. I'm just writing stop for your sake, but you don't need to write stop on the test. You can just give me the peptide sequence. So that is, like I said, somewhere around 3-ish, maybe 3.5 marks out of the whole thing. We are going to talk about mutations, and that will probably take up the other maybe 1.5 to 2 marks for this kind of question. Like I said, I haven't written the exam yet, so I don't exactly know, but that's usually what I do. And I can't see myself doing too much different, maybe, you know, tweak it a little bit. So we will see more of these questions. And you may have noticed if you go on to Moodle, under the lecture notes, I've put a like a final exam study guide. And so you can take a look at that study guide, and I have one of these kind of questions there in it with a solution. So I encourage you to, you know, print that off or work on the question and you know, work on the whole thing and then look at the solution. Okay, and hopefully you can get this right. And like I said, this will be at least about 5 marks on the final exam. and probably other questions you'll find around these kind of concepts as well. So hopefully it'll be easy. I'm hoping this is easy marks. Like I said, final exam, listen to me very carefully. This is a promise. There will be something exactly like this in the final. And so it's important that you are able to do it. And so like I said, just take a look at that practice question, and hopefully that will help you with this. Okay, so like I said, I want to talk about what happens after the messenger RNA is made, because there's all this modification going on. So you have that messenger RNA, and there's a couple of things that are added on right away in eukaryotes. One is something called a 5' cap. So what is a 5' cap? Is at the 5' end, right? So hopefully that makes sense. And a cap, I guess you can kind of think of it as like a hat or a hard hat or something like that. And that's actually a pretty good analogy, right? Because you can see that its functions are to protect it, right? So think of it as a hard hat, and a hard hat that has a label on it that says, I need to get out of the nucleus, so that those nuclear pores that we were talking about a while ago, they don't just let things go in and out. They are like security guards. There are rules of engagement, rules that say, hey, this goes in, and hey, this goes out. So messenger RNA is definitely something that needs to get out. So that's kind of the second point that the 5' cap is doing. And it also helps with ribosome binding. Ribosomes kind of need little, they're going to grab onto the RNA and just kind of an extra handle on there. At the other end, there is a poly-A tail, and this is mostly to protect it from degradation. So you can see it's basically a whole bunch of A's. So adenines, and maybe 50 to a couple hundred is pretty typical. And this basically prevents degradation. So RNA is a very sensitive molecule, and it really likes to degrade at the 3' end. And so this is kind of like a telomere in that it just offers some protection. It will degrade eventually. So the more A's that are put onto there, the longer that messenger RNA will stick around. Some messenger RNAs are only around for minutes. And protecting them this way is just kind of the main thing here. I have a picture of the 5' cap, so you can see what it looks like. So what we're looking at is 3 phosphates, 1, 2, 3. We've got another ribose sugar here, and then we've got a guanine, and the guanine has an extra methyl group on it. So just a little bit of chemistry for you there. So like I said, it protects it and signals it for nuclear export. So it needs to get out of the nuclear pore, and that's very, very important. And I guess I said all that already. Okay, so one other thing that happens with our free messenger RNA, so we've added on our cap, we've added on a tail, and now we're going to do something called splicing. So if you take a look at this gene here, we've got these things called exons and introns, okay? And it turns out the introns are cut out. So they kind of have, the naming is a little bit confusing. Again, I blame geneticists. I don't really like their style for naming things. So exons are expressed. So that means that's the part of the gene that is read by the ribosome. Introns is short form for interruptions, right? So these are interruptions to the gene, and those are cut out. So I know this is like crazy. So why did you go through the bother of doing all this, and then we're just going to cut out half of the gene anyway? And talk about that in a moment. But it's something that's done in many, I'm not sure if it's all eukaryotic genes, but the majority of them for sure. They're cut out. And so introns and exons, like I said, just complicating things for all the biology students, unfortunately. So how is splicing done? So splicing is done by something called the spliceosome. So what is the spliceosome? It's this big complex of proteins and RNA. And what it does is, you can see in this diagram, it's going to recognize those exons. So it binds the exons, and it can cut them out. So there we go, it cuts it out. So there goes the introns, that's going to be discarded. And this is our messenger RNA with it, which is just the exons left over. So take a look at this. We have another type of RNA. So we're talking about messenger RNA today. Messenger RNA is what is coding for the proteins, and small nuclear RNA is what is found in a ribosome. So small nuclear RNA, it was named because it's found in the nucleus and it's small. Sometimes you see this term here, which is slightly different, small nuclear ribonucleoproteins. So you can see it's protein and RNA. complex together, right? So small nuclear RNA, I'll write a definition here. So this has structural and catalytic roles in spliceosomes. And what's a spliceosome? A spliceosome is a complex of proteins and RNA that are involved in splicing. Now, what is splicing? Splicing is where the introns are cut out. Okay, so lots of new words for you to bring home and talk about at the dinner table today. Yeah, so two more types of RNA today, messenger RNA and small nuclear RNA. So I wanted to point out one other thing here. So notice I said it has structural and catalytic roles, okay? I want to just talk about that word catalytic for a minute, right? What does catalytic mean? Catalytic means something to do with an enzyme, right? So I had mentioned before that enzymes are usually made out of proteins, and here is one of those exceptions. So sometimes enzymes are made out of RNA. So the term for this is ribozyme. So one more definition for you. So what's a ribozyme? A ribozyme is an RNA enzyme. So ribonucleic acid, which is RNA, and then enzyme. And there's been a whole bunch of these that have been discovered. Usually enzymes are proteins, but once in a while we find these ribozymes that are not proteins and they're involved in other processes. So one process, for example, is splicing, which we just talked about now. And actually the other process here is something we're going to talk about next lecture, which is the catalysis of new peptide bonds at the ribosome. So RNA is a huge part of the ribosome and is actually involved in the catalysis reaction. So kind of a little side there, another definition for you. Okay, so I had asked the question, you know, what is going on with those introns? What is the point? And there's a lot that we don't know about introns, but we have made some observations in some areas where they are useful. So sometimes these exons, they're different domains of a protein. So what do I mean by domain? I mean kind of these folded sections. So you can see we've got domain 3 here is coded by exon 3. We've got a second domain coded by exon 2. So this is a way for proteins sometimes to, you can have one gene and you can actually get different proteins out of this depending on which exons are actually used. So it's kind of interesting. We've seen this a number of places in the biological world. The best place that this has been investigated actually is part of your immune system. I know this is talking about a gene called troponin, and I'll show you how this works. So the term for this is called alternative splicing. So take a look at this and I'll talk about how this works for the immune system in a moment. But basically, the gene is read by the RNA polymerase, and you get a big piece of messenger RNA, and then you get a whole bunch of RNA. And the splicing can occur, and the splicing doesn't necessarily have to take all the components, right? So you can see in the case here, this option here, the splicing is taking components 1, 2, 3, and 5. Over here, it's taking components 1, 2, 4, and 5. So you can actually get different versions of a protein, which is useful in some cases. So like I said, the biggest use and the best understood case of this kind of phenomenon is in your immune system. So let's talk a little bit about the immune system here. The immune system, a big part of the immune system has something called antibodies. So this is what an antibody looks like. It's kind of a Y-shaped protein. And kind of the relevant thing about an antibody is it has this section here. You can see it says here, this section is what is going to recognize something called an antigen. So what is an antigen? Antigen is like something that's going to invade your body. So it could be a virus protein. It could be a surface structure on a bacteria. It could be a toxin. So these molecules, these proteins, are designed to recognize foreign invaders. So I'll show you a little example here. Here's a little antibody, and there's a virus, and the antibody recognizes the virus. So let's say maybe this is let's say this is chicken pox, right? And you get infection with chicken pox, and the antibody recognizes the virus, and then what happens is immune cells recognize the antibodies, and they're going to destroy it by endocytosis. And so if you've been exposed to some diseases, sometimes we get memory, and the second exposure is going to be stronger and faster, and you may not even know you were exposed to the virus. So this is what we're trying to do, obviously, with making a vaccine. So the thing is about these antibodies is that there are way more antibodies out there than our genome can code for. Our genome codes for about 20,000 proteins. There's literally about 1 billion antibodies out there. And so how do they do this? We only have 20,000 genes. Well, we do this with that alternate splicing. So you can see here's the region of the genome for antibodies, and it has a whole bunch of exons. And you get all sorts of different mixing and matching going on. And so you get all sorts of different antibodies to recognize many different potential invaders. And it's kind of a really cool process. So I thought I'd share that with you. But the main takeaway lesson from today is to know what's going on with transcription, okay? Know the processes behind that and some of the quirks that are different between prokaryotes and eukaryotes. So speaking of which, I made a table for you, it looks like. So what is going on in prokaryotes? Remember I said in prokaryotes, they have 1 RNA polymerase. In eukaryotes, there's 3, although if you're a plant, you might have 5. I'm not sure about some of the other weird things out there like fungi and protists, but I wouldn't be surprised if there are other versions of transcription in some of those other organisms. What about transcription factors? So bacteria, no in bacteria. Archaea, yes, I think it's more like a sometimes though, and in eukaryotes, yes. So shows you that sometimes archaea are a little bit more like eukaryotes than bacteria. What about splicing? None ever discovered yet in bacteria. You never know. There's some that has been discovered in archaea and then very, very common in eukaryotes. Plus, you're going to get the capping and tailing in eukaryotes because we have a nucleus and we have to get out of that nucleus, and things are just so much more complicated because we're such large complex organisms. So I think that is actually the end of today's lecture. Usually it takes a little bit longer because I'm spending more time on the whiteboard, but that's fine. We'll finish a few minutes early, and I guess on Wednesday we're going to talk about translation, and I also want to talk about the lab exam on, wait a sec, today is Wednesday, sorry. Friday, we're going to talk about translation. And I'm hoping that tomorrow, if I don't get it out tomorrow, then it'll be Friday. I'm going to give you guys information about the lab exam, which is 1 week from tomorrow. So watch for that information. All right, have a good night. We'll see everyone on Friday.